Hello, my name is Sakshi Sandhir,
Hi, I'm Priya Mathy.
and thank you for joining us as we discuss our research project that focuses on exploring the efficiency of differentially substituted 4-thiazolidinones on gastrointestinal stromal tumor cells. So a little about us. I, Sakshi, am a third-year pre-medical student, and I'm currently pursuing my bachelor's in general biology.
And hi, I'm Priya. I'm also a third year pre-medical student, currently a medical assistant for an OBGYN practice, which has inspired me to pursue oncology research.
So let's get started. As we dive into the fascinating world of cancer research, we will be focusing our discussion on gastrointestinal stromal tumor cells, otherwise known as GIST cells. So GIST specifically originate from a cluster of cells that rise from a distinct type of nerve cell present in the digestive organ's walls. So for our project, we wanted to focus on the potential of 4-thiazolidinones as anticarcinoma agents in relation to our GIST cells and aim to explore potential therapeutic efficiency in tumor treatment. So the organic compounds that are used in our study include Bromo, Trifluoromethyl, Chloro, Fluoro, Methyl, and Phenyl substituents of the 4-thiazolidinones. For these compounds, we explored each with two specific arrangements, meta and para substitution. So meta refers to a molecule with substituents positioned at the 1 and 3 positions of the compound, and para describes a molecule with substituents at the 1 and 4 compound. So let's continue our discussion by walking through our experimental procedures. So firstly, the GIST cells were cultured in the Dobecco's modified Eagles medium. So this culture took place in a controlled environment at about 37 degrees Celsius and 5% carbon dioxide. We then plated approximately 50,000 cells per well in 6 well plates, and these plates were then incubated for 24 hours, which allowed the cells to establish and grow. After the initial incubation period, these GIST cells were then exposed to various concentrations of our previously mentioned organic compounds, ranging from 0 to 400 micromolar. As the cells were closely monitored after 48 hours of exposure to the compounds, visual observations were conducted under a microscope in order to assess their status. So for further inspection, cells were cultured on glass cover slips and then observed using Nomarski optics to visually capture the effects of the compounds.
So let's dive into the findings of our study. Our results indicated that 4-thiazolidinones exhibited both anti-proliferative and toxic effects on GIST cells. So it is essential to prioritize the review of our controls. The absence of a control makes it difficult to determine the effects and the outcomes of our organic compounds due to their exposure, as opposed to being due to some other variable. On this slide, we can observe our control GIST for 300 micromolar. And on this slide, we can observe our control GIST cells on 400 micromolar. So we observed a dosage-dependent response. At a lower dosage of 300 micromolar, compounds such as P-bromo, P-fluoro, and P-methyl demonstrated significant effectiveness in inhibiting cell proliferation. However, at a higher dosage of 400 micromolars, a different set of compounds emerged as more potent inducers of cell death, notably M-bromo, P-bromo, M-trifluoromethyl, P-fluoro, and phenyl exhibited higher efficiency in inducing cell death. It is important to note that precipitation and crystal growth pose challenges in accurately quantifying cell growth. Interestingly, precipitation increased with higher concentrations of the compound. Overall, these results provide valuable insights into the interactions between 4-thiazolidinones and GIST cells, paving the way for further exploration in the field of cancer therapeutics. Our project is currently underway with ongoing plans to assess the prolonged effects of 4-thiazolidinones in gastrointestinal stromal tumor cells, including examining how different concentrations influence cell growth over an extended period of time. Additionally, we aim to potentially investigate the effects of these compounds on other GIST cell lines. We extend our gratitude to Fox Chase Cancer Center for providing GIST cells essential for this research, and Dr. Ingersoll for his guidance and support throughout the project. Thank you.